Hello students, welcome to my YouTube channel, Study of Life Sciences. This is the first lecture on YouTube. Here I am going to start the basics of cell biology, and today we will deal with some basic concepts regarding cell, regarding prokaryotes and eukaryotes and their differences. I hope this lecture will be helpful for you in different exams. This topic will be helpful for CSIR, UGC, NET, NEET exam and all other competitive exams. Here we are going to start our first lecture, that is basics of cell biology. Here is our first slide. What is cell? If we are talking about the cell, here you can see cell. Cell is the basic structural functional unit of life. I hope everyone knows that cell is structural basic functional unit of life. Cell is the smallest unit of life. Cell is microscopic in origin. What do you mean by microscopic? That cannot be seen by our naked eyes. Discovery of cell. The first cell was discovered by Robert Hooke. But the cell is called as dead cell because that was discovered from the cork. And second thing, the living cell was discovered by Anton von Leeuwenhoek. Anton von Leeuwenhoek. This is the important point. Living cell. Who discovered the living cell? That is Anton von Leeuwenhoek. And now we will come to the third concept, cell theory. Cell theory was given by the botanist Moritz Schleiden and a zoologist Theodor Schwann in 1839. Rudolf Virchow then came, another scientist, in 1855. What he tell in 1855? Robert Virchow came, defines omicellula and ecellula. Omicellula, ecellula. It was basically a cell theory. He defines that all the cells came from pre-existing cells. It means that all the cells came from the old cells that were already present in the environment, pre-existing means primitive cells. And he gave some another concept, that is about DNA and RNA. He concluded with this matter that he told that DNA and RNA as a genetic material which is present in the nucleus, of a cell, and that is transferred from one offspring to another offspring. When there is a cell division, when the cell divides, if it gives asymmetrical division, it gets into two halves, and one house from mother and one house from father, and it combines again, it makes some zygote. After that, the genetic material is contained in this cell. I hope this may be clear to you. Now we can come to the cell shape. Generalized shape of cells is round, spherical or irregular. Round, we can say that also spherical and irregular. Another thing is that microbes have oval shaped cells. Animals have rectangular, spherical, and plants have cuboidal, spherical and rectangular. Smallest cell in the living environment, that is mycoplasma, which has a diameter of 0.001 nanometer, and largest cell is the egg of an ostrich. It is a single cell. You can conclude egg. If we are talking about the egg, it is a single cell. We are talking about here the single cell. Single largest cell, smallest cell is mycoplasma, and largest cell is ostrich egg. I hope this slide may be clear to you. Now we can come to the next slide. We are discussing the shapes of the cells. Round. If we are talking about the round shape, oocytes means female sex cells from which the ovum is made. It has a round shape. Liver cells have oval shape here, and Squamous tissue squamous tissue cells have flattened shape, and there are some cube shaped cells, that is called cuboidal, some columnar shaped cells, they may be columnar cells. Elongated cells are present in collision. Elongated cells are present in collision, you can consider it. Star shaped cells are also called as natural killer cells. Star shaped cells are natural killer cells, and they are star shaped. And then came to the another class of cells, that is cylindrical. Cylindrical cells are present in skeletal muscles. Now we are talking about the disc shaped cells. disc shaped cells disc shaped cells are found in rbcs. If we are talking about the rbcs if we are talking about rbcs, they have usually, as I have told you, that there is a disease that is known as sickle cell anemia. Anemia. They have sickle shaped cells, just like dumbbell. They have sickle shaped cells. And one more concept about this those rbcs, they are non-nucleated. They does not contain nucleus. Initially they may have nucleus, but in later stages they does not have nucleus, they are nucleusless. And one more concept about muscle cells. Muscle cells also does not contain nucleus. Muscle cells also does not contain nucleus. Here we are talking about the muscle cells and RBCs does not contain nucleus. Without, it means absent nucleus. I hope this may be clear to you. They are without nucleus. If we are talking about the general animal cell, the cell membrane, that is also called the plasma membrane. That is also called, here we are talking about the animal cell. This is the plasma membrane, outer membrane. It is also sometimes cell membrane, called cell membrane. Inside the plasma membrane is cytoplasm, and in the cytoplasm there are different organelles: mitochondria, nucleus, DNA, endoplasmic reticulum, lysosomes, ribosomes, Golgi apparatus. I hope later on these organelles will be must discussed in the later on lectures. Here we are giving the general idea about these organelles. If we are talking about the mitochondria, everyone knows that powerhouse of the cell from which the ATPs are generated. Nucleus, inside the nucleus there is DNA, genetic material, and in virus and all that there is RNA. And endoplasmic reticulum. Endoplasmic reticulum helps in the transport, retrograde and reverse retrograde transport. And if we are talking about lysosome, these are called suicidal bags. They maintain the cell structure. When the cell division increases, that leads to the disease, that's cancer, and they help to maintain the cell division. If we are talking about the ribosomes, the protein synthesis factories, they may be usually present on the endoplasmic reticulum, and they may sometimes free in the cytoplasm. And Golgi apparatus, these are the main organelles that help in the transport. Golgi apparatus and endoplasmic reticulum are the main cells that help in the transport. I hope this may be clear to you. Now we can come to another concept, that is plant cell. The major difference between plant cell and animal cell is that plant cells have chloroplast, plant cells have cell wall. These are the two major differences in between the plant cells and animal cells. Here is cell membrane also, plasma membrane, which is called plasma membrane sometimes. This is nucleus, this is chromosomes, cytoplasm, and some other organelles, fluid space. And one more thing that is present in the plants, that is a large sized vacuole. Large sized vacuoles are present inside the plant cell. And one more thing about these cells, here we are talking about the chloroplast. and mitochondria. Where is mitochondria? Mitochondria is not shown here. Mitochondria is also there. Mitochondria. These are and nucleus and nucleus. These all are double membrane organelles. Double membrane organelles. Double membrane organelles. Double membrane organelles. Here we can discuss double membrane organelle. If this is the nucleus, it has inside another, and inside there is nucleolus. Nucleolus. And if we are talking about the mitochondria, just like this is also double membrane. And the space in between this is called intermitochondrial space, and this space is called nuclear space. Nuclear space. I hope this may be clear to you. This is called intermitochondrial space. Intermitochondrial space. Now we can come to another concept, that's organisms. Organism may be single cellular. Organism may be cellular or acellular. Cellular, which are made up of different types of cells, and acellular, which does not have definite cell shape. Usually, viruses and viridis are included in acellular organisms. Viruses and sorry, viruses and viridis are included in this acellular organisms. What is here minus pm? Minus pm means they does not have plasma membrane, and acellular organisms have particular cell structure and they contain plasma membrane. On the basis of the nucleus, the cells are further divided into two categories. One is eukaryotes, they have proper nucleus, and another is prokaryotes, they does not contain nucleus. Furthermore, the eukaryotes are further divided into two categories. It's multicellular organisms and unicellular organisms. And prokaryotes are also divided into two categories, archibacteria and eubacteria. Archibacteria and eubacteria. Here you can see the archibacteria. Here is the minus sign. The eubacteria and archibacteria are classified on the basis of 16 srRNA. Ribosomal RNA. Eubacteria contains 16 srRNA and archibacteria does not contain 16 srRNA. I hope this may be clear to you. Now we can come to the multicellular organisms. Multicellular organisms here is that they are heterotropous and they are autotropous. Autotropous means they make their own food. They make their own food. But heterotrophic organisms does not make their own food. They are dependent upon autotrophs. And one more concept, heterotrophic organisms use organic carbon and autotrophic organisms use inorganic carbon. This is one of the main difference between autotrophs and heterotrophs. And one more thing, now we can come to the unicellular organisms. Unicellular means they are single-celled organisms. They may be photosynthetic, which can do photosynthesis, and they may be non-photosynthetic organisms. Photosynthetic organism is Euglena. And non-photosynthetic organism is Paramecium and Amobia. But you can remind it, Euglena, Paramecium and Amobia, all the three are single cellular organisms. They are made up of single cell. I hope this may be clear to you. Now we can come to the next slide. Multicellular. As I have told you, multicellular organisms are made up of more than one cell. They are heterotropous, they are autotropous. They can use carbon dioxide as inorganic carbon, and they can use organic carbon. Further, autotrophs are further classified into two categories, algae and land plants. And heterotrophic is further classified on the basis of mode of nutrition. mode of nutrition. Here you can consider it mode of nutrition. Ingested animals and the animals those are dependent upon dead and decayed matter such as fungi and bacteria. They are showing a process that is known as adsorption. Adsorption. I hope it will be discussed very soon. Adsorption. And the major difference between ingestion animals and dead matter decaying animals is that they can take directly food through their mouth ingest, and they are dependent upon the dead and decaying matter. They first secrete some enzymes. Here I will show some a little bit example. If this is a dead and decayed matter, or waste material which is thrown out of our houses, waste materials, bacteria came here. This is not basically a bacteria, but I am showing in the diagram. Can secrete some enzymes, that gets adhered. Adhered. Adhered means they can combine, they can fuse with this waste material, and the secreted enzymes will adsorb to the waste material, and they can make their nutrition possible. And now we will talk about the differences between eukaryotes and prokaryotes. Prokaryotes and eukaryotes. As you know, prokaryotes came first, and after that eukaryotes came into the existence. If we are talking about the nucleus is present in eukaryotes and absent in prokaryotes. Origin of the eukaryotes. Is 1.5 origin of the eukaryotes is 1.5 million years 1.5 million years ago. 1.5 million years ago. And origin of the prokaryotes is 3.6 million years ago. Size. Prokaryotes are smaller in size and eukaryotes are large in size. Surface area volume ratio is high in prokaryotes and low in eukaryotes. Double membrane organelles. The organelles which are double membrane are absent in prokaryotes and present in eukaryotes. And one more thing, we will talk about the ribosomes. We'll talk about the ribosomes. We will talk about the ribosomes. I hope this may be clear. Prokaryotes contain 70s ribosomes. The 70s ribosomes are made up of two units. One is 50s and another is 80s. Another is 30s. These two subunits combinedly make the 70s ribosomes. I hope this may be discussed in detail in later on lectures. And one more thing, the eukaryotes are made up of 80s ribosomes. It has also two subunits. One is 60s subunit and another is 40s subunit. These two combine together and form the 80s unit. One more concept here. Double membrane organelles. Those are absent. Double membrane organelles which are absent in prokaryotes but are present in eukaryotes. And those double membrane organelles are nucleus, mitochondria, and chloroplast. These are the organelles, double membrane organelles which are present in eukaryotes. Now we can come to the next slide. Here, difference between bacteria, animals and plants. If we are talking about the structure, cell wall, here we can take the cell wall. Cell wall is present in bacteria but it's made up of peptidoglycan. Now we are talking about the animals, they does not contain cell wall, and plants, cell wall is present and is made up of cellulose, enzyme cellulose, plasma membrane. Now we are talking about the plasma membrane. As you have seen, plasma membrane is present in bacteria, plasma membrane is present in animal cell, plasma membrane is also present in plant cells. Flagella. Flagella is present in bacteria, flagella is present in animal, and flagella usually is absent in the plant cells, but some sperm cells may contain flagella. Before going to discuss more, I will tell you some basic concept about flagella. Flagella usually, if this is a bacterial cell, this is a flagella. Usually this is for the movement. This is for the movement of cell. Movement of cell. Usually plant does not move from one place to another place. That is the reason they does not contain flagella, and it may be present in some sperm cells, in some sperm cell, because sperm has to show the movement, otherwise it will not reach to its destination. Flagella is present in bacteria, present in animal, and absent in plant cells. Some sperm cells may contain flagella. Microtubules are absent in bacteria, present in animal, and present in plant cells. Centrioles are absent in bacteria, present in animal cells, and present in plant cells. Animal cells, usually if we are talking about the humans, centrioles are not present in the female. Male centrioles take part in the cell cycle. True nucleus. True nucleus is absent in bacteria, present in animal cells, present in plant cells. Chloroplast. Chloroplast is absent in bacteria, absent in animal cell, only present in plant cells. Chromosomes. Chromosomes is absent in bacteria. Instead of chromosome, it contains chromatid. Present in animal cell, also present in plant cell. I hope this may be clear to you. We can come to the next slide. If we are talking about the ribosomes, as I have told you that those animals have ATS, they also have ATS, they are also eukaryotes. Bacteria are prokaryotes, they have 70S ribosomes. Vacuoles. Vacuoles are absent in bacteria. Animal cells may have small vacuoles, but plant cells have large vacuoles. Peroxisomes. Peroxisomes are absent in bacteria. Peroxisomes are present in animal cell, and peroxisomes present in plant cells also. Glyoxysomes, absent in bacteria, absent in animal cell, and present in plant cells. Mesosomes, one of the most important feature of bacteria. Bacterial cells contain mesosomes. Mesosomes show the cellular respiration. Here you can see cellular respiration. It is absent in animal cells, it is absent in plant cells. Here the cellular respiration is done by mitochondria. Here also it is done by mitochondria. And mitochondria is absent in bacteria, present in animal cell, also present in plant cells. Now we can come to the next slide. This may be our last slide today. Cell composition. Cell composition. Here if we talk about the cell, this is a cell. In the cell, first off I'll give you some basic concept. K positive concentration is high, Ca2 positive concentration is low, Na positive concentration is low, Mg2 positive concentration is low, and there is only one Cl negative ion. Mostly these ions are present inside the cell. Less amount of the positive charge is inside the cell. Here we are considering there is a less amount of positive charge. We may consider it that inside is a negative charge. I hope this may be clear to you. And outside must be positive charge. That is the reason plant cell animal cell, both cells have outside positive charge and inside negative charge. I hope this may be clear to you. Now I am going to tell you about the cell composition. Cell contain different components. First of all, we can talk about the amino acids. Amino acids made up of carbon, hydrogen, oxygen, nitrogen, and sulfur. Sulfur. If we are talking about the sulfur, sulfur is present in cysteine, that is the amino acid, and selenocysteine. Seleno, selenocysteine, that is the 21st amino acid. 21st amino acid. 21st amino acid. And one more thing, second concept, nucleotide. Second concept, sorry, nucleotide. 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 Nucleotides are present in the cell also, cell composition. They may contain carbon, hydrogen, oxygen, nitrogen, and phosphorus. Amino acids does not contain phosphorus. Does not contain phosphorus. Now, fatty acids. Here, fatty acids. Fatty acids are made up of hydrogen, oxygen, carbon, and phosphorus. These four components. I hope this may be clear. Now the last, carbohydrates. Carbohydrates. This is the last one. CHO. Carbohydrates. Now if we can conclude this, we can sum up this. Here the nutrients, here the element, that is carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphorus. This is your most important thing that is present inside the cell. We can tell it a chonsp. I hope this may be clear to the you. These are the basic things which are present inside the cell. 98% of the cell, and 70, if we are rest of this, 70% is water. That is the solvent of the cell. That is the solvent of the cell. Now we can come to another concept. Cell. If there is a cell without cell wall, or plasma membrane. Cell without plasma membrane or cell wall is called protoplast. Here you can see cells without plasma membrane and cell wall is protoplast. Protoplast may be living protoplast and non-living protoplast. Living protoplast is cytoplasm and nucleus that is present inside the cell. And in cytoplasm there are organelles, as I have told you before. They may be single cell organelles. Single cell organelles, lysosomes, and double membrane, I have told you it already, mitochondria, nucleus, and chloroplast. These are double membrane organelles. Now, if they are non-living, if there is non-living protoplast, this may be the important point. Non-living protoplast is ergostic bodies. In the next lecture, we will discuss about these ergostic bodies. I hope this may be clear to the ergostic bodies or non-living protoplast. Living protoplast, cytoplasm, nucleus, in cytosol, the whole cytoplasm, organelles. They may be single cellular organ, lysosomes. They may be double membrane organelles. This is all included in the living protoplast. Now I have told you that if the positive inside charge, we will maintain this pH inside the cell. Inside the cell, slightly basic, or we can say alkaline, 7.2 to 7.4. It is considered in general 7.2 to 7.4. And one more thing, ribosomes which are attached to the plasma membrane, or sometimes which are attached to the endoplasmic reticulum or suspended freely in the cytoplasm. Ribosomes are without membrane. They does not have membrane. One more important point, ribosomes does not have membrane. And one more thing that I have clear at first, more positive charge outside the cell. Here you can see more positive charge outside the cell. I hope this all may be clear to you. Now we will meet in the next lecture If you like this video, do like and share and subscribe this channel. Now we will meet in the next lecture of plasma membrane and transport across cell membrane, transmembrane protein passes. Thank you for watching this video.